what is the difference between research use peptides that say not for human use and compounded prescriptions from a 503A compounding pharmacy or a 503B compounding pharmacy, right? So when you're doing research use only peptides, you have no idea where they're coming from. You do not know if there's contaminants in it. It is not intended to be used for medical use, right? And that's how they skirt the rules around the FDA. The FDA only imposes their will on intended use. So when you literally say it's not for human use, then they're kind of skirting the line on having to follow any of these regulatory practices for delivering good quality products to people. But people still use it. And it's a lot of people using it, right? We have data to show how many people visit these sites based off of all the tech that exists today, and it's wild. So like the difference between research use and then we have compounded prescriptions, right? The compounded prescriptions, one, have to be written by a physician. There's medical supervision there. Their pharmacies have to follow USP guidelines. And at the end of the day, if you're following USP guidelines, the compounded prescription is tested for potency, purity, endotoxin testing to make sure it's not going to harm somebody. And what goes out of the door to the patient is actually what's in the vial, right? That is the biggest thing that I think most people are missing, is that this stuff is research use only peptides are not intended for human use. You have no idea if you're getting chalk powder in a vial, right? And they have to reconstitute it. Just the fact that they have to reconstitute this without any instructions, because they can't even give them instructions technically, because once the research use manufacturer tells a patient or a client, whoever's buying their stuff, this is how you should reconstitute it, they're breaking the law. Because why would you have to tell them how to reconstitute it if you're doing research? Right. So some of these people don't even know what they're doing when it comes to reconstituting. Are they taking tap water and putting it in here? Like, who's guiding people on this journey? And it's so bad. And I wish there was somebody looking at the amount of sepsis that's happening or the infections that are happening when people are using these things, or like just high doses leading to issues.
Yeah. And like, even if the worst case scenario is you get contaminated product, sepsis, that kind of thing, even if it's not that, low levels of exposure to endotoxins, like these are bacterial byproducts. Lipopolysaccharides is a big one, LPS, that there's standards for human consumption that have to be followed, that doesn't have to be followed for research purposes. Best case scenario is that you're inducing small level chronic inflammation every time you inject.
Yeah.
Right. That's best case scenario for this. When we do studies on people and we want to check inflammation, we inject them with LPS.
Yeah. The biggest difference between if there is LPS, right, is because sometimes people ask me, so how do compounded prescriptions prevent that from happening? Like, how do you guys ensure, if as a compounding pharmacy, that you don't have those endotoxins in your final product? One, it's following the process and making sure the mixing and all that's happening in an ISO class hood, right? That's why you see the hoods in a compounding pharmacy. So following proper gowning procedures, not introducing bacteria into the situation. So there's a lot of rules that need to be followed, even when you're handling these chemicals. And say worst case scenario, there is something that gets in, right? Your end product is sterilized by a couple of different forms, depending on what the product is. One could be autoclaving. Like, you literally heat it up to a certain temperature where all bacteria and viruses die, as long as it doesn't affect the byproduct, right? The other one is it can go through a filtration mechanism. So when you're filtering these things out with sieves that are so small and capture viruses and bacteria, you're getting rid of all this stuff. So your final product goes through all these sterilization techniques that it should be very pure. And on top of that, that product has to go out for testing. So you're testing for potency, you're testing for purity, you're testing for endotoxin, and making sure that there's no harm that can be done by getting this thing out the door, right? So that's the biggest difference between research use only peptides and compounded products, if anybody's wondering.
All right.